All right, I'm Robert Green, and thank you for staying this late in the day. Now that all the problems have been solved, we get to talk about integrating them into clinical pathways. And I would remind you that although I know that the early planes will be tempting, I will remind you that not only is this going to be exciting, but the panel after this has Sharon Terry's husband in it, so she'll be noticing who's here and who leaves early. So we're going to structure this. We're going to try to keep this moving in a very lively way and structure it in the following way. I'm going to make about 2 minutes of remarks and introduce myself. And then each of the speakers, including myself, is going to give 5 minutes on what we think is a salient issue related to clinical integration, and ending with a one-line statement of a problem that we would like to see fixed. And then we're going to go back through and to each of the 5 and give a one-line solution that we've come up with for that problem. And then we're going to open it up. And if we do it right, we will have a good 12 to 14 minutes of discussion, even if we shorten it by 5 minutes. So I'm going to tell you 1 minute about myself because I don't appear in the program, but that's okay. I'm new to genetics, and I was originally trained as a neurologist, trained in public health, and now have retrained in medical genetics. And I'm very excited to be running a program in translational genomics and health outcomes, basically comparative effectiveness studies and outcome studies and how we apply genetics and genomics to real-life medicine and social situations. And I'm at Brigham and Women's Hospital and Harvard Medical School, and if you want to reach me, my email is easily available on the web. So that's my introduction. So now I'm going to ask our speakers to give their 5-minute shtick, and I'm going to ask them to stick with 5 minutes or less as they begin. So, Greg, would you like to start us off?
Sure. My name is Greg Feero. I'm a family physician, as we found out earlier. I'm also a special advisor to the director for the NHGRI on issues in genomic medicine. My primary care world is actually in Maine, in a rural area in central Maine, and I practice in very much a community-based setting. So I frequently get to see sort of both ends of the spectrum of the promise versus the reality of integrating genomics into healthcare. So I'm going to take an anecdote from I had a very interesting day this last Tuesday. I was actually going to use 3 bits, but to shorten things up, I'm going to just talk about one of the sort of anecdotes that came up on Tuesday, March 6th for me. And I think it's an issue that's going to become increasingly challenging as we move forward with more applications in genomics that are relevant to healthcare practice. So I am in charge at our residency of teaching our residents about evidence-based medicine and thinking about how to become more effective consumers of healthcare. And certainly they, by dint of my background, get a little more genetics than the average family medicine resident as examples of the challenges of becoming an educated consumer. So this past Tuesday, we had 3 sessions in the afternoon where I was charged with helping them to become better at ascertaining and rating guidelines for care that would allow them to sort of take a more evidence-based approach to clinical problems. So we set up 3 brief clinical vignettes, and then I asked the residents to break down into small groups and to use several reputable sources to look up guidelines that would help them manage those patients, and then rate the guidelines in terms of the quality of the guideline, and then talk about whether they would use this guideline to change their management or not. And I slid in a couple of genetic and genomic examples, of course, in there to just see what would happen. And one of the things that really was striking that came back was that there is a real profusion of guidelines at this point in time, both for genetic and genomic topics and for the rest of healthcare. For one of the simpler vignettes, we came up with 23 guidelines, variably evidence-based, and for at least one of the vignettes, we came up with a number of conflicting guidelines. And that leaves the primary care clinician in a bit of a quandary. Further, those guidelines serve as a basis for emerging metrics of quality of care and pay for performance, and it will really leave individuals in a quandary when they are subjected to sort of varying criteria for quality as they move from institution to institution, depending on that institution's flavor of the month of guidelines that they decide to adopt as a quality metric. And so the problem, as I see it, is one of the problems we're facing is a profusion of guidelines. I didn't even use the word evidence in my talk, which is unusual for me, but a profusion of guidelines as we move forward, variably evidence-based. I think we need to think about ways to begin to become more homogeneous or aligned in our adoption of these guidelines.
Excellent. Thanks very much. So, Greg, is it fair to say that your problem is too many guidelines, and your solution is? Oh, well, no, we'll come back to the solution. That's right. Okay.
Has had in his life with dealing with certain social and work and educational issues. So it brought up a very complex conversation about was this the same exact kind of a phenotype that one would see in the fetus? Would it be less severe, more severe? Did they care? And so it's just an idea about how we're getting information that we really have not had a lot of experience on how to deal with it in the past. And this is going to be one of the big obstacles to adoption in the current pathway. Right on time.
Perfect timing. And so, Thomas, is it fair to say that your problem is information of uncertain significance on chromosomal microarray in amniocentesis and before too long on DNA from maternal blood? And you're going to give us a solution to that, so that's going to be awesome. And Deborah, please introduce yourself and tell us about your problem and your solution. Not your solution yet, your problem.
Okay. My name is Debra Hein, and I'm a little different than most of the panel. I'm not a physician. I'm an attorney by training and got involved with genetics when my second daughter was diagnosed with spinal muscular atrophy at 8 weeks and died at 10 months. So Chris and I, my husband, after Claire died, we looked out into the sort of landscape of non-for-profits and saw a lot of groups working on treatment and cure. And we didn't see anybody who was working on the role of carrier screening or prevention. And my husband, I think, sort of sums up our mission very neatly when he says, no one should find out you're a carrier of SMA by having your child diagnosed at 8 weeks. And that's really the foundation of what we did. So then we started a foundation, and we thought we were going to do something really simple because a carrier test existed. It had been used by the SMA community for a long time. So we thought, oh, we're just going to get involved in policy, and we're just going to get the test offered. We're going to try to bring it into a clinical pathway. Well, 7 years later, we're here. It wasn't so easy. We're a group that has conflicting guidelines where we have ACOG saying it should only be recommended to those with a family history or those requesting a test. And we have ACMG endorsing all populations should have carrier screening for SMA. So I'm not going to talk about that because that's Greg. But my problem that I started to think about, I sort of had to, I'm going to cheat a little, is the first thing that Sharon brought up on the agenda. So the appropriate uses of genetic testing. And so my first question is, well, who determines that? And I think that is sort of the starting point in a lot of ways. And right now it is these practice guidelines. And I would argue that we need to create a pathway to actually get to the point as to who brings a test into the clinic. And I don't know that guidelines by sort of siloed, discrete groups is the right way to do it. And so sort of my problem is, how do we create a pathway that actually brings the testing into the clinic? Would be the first thing. And then my sort of cheat is the cost-benefit analysis and some of the things that go around what is the benefit to reproductive testing. And I think it's one of these elephants in the room because what is the benefit? Reproductive testing, oftentimes the benefit is you can choose to bring a child into the world without disease. It's a benefit that we don't like to measure. We don't like to talk about abortion. We don't want to talk about PDG. We're not going to go around and collect that kind of data, understandably. So then how do we figure out what the benefit is and how do we give that the weight that it needs to be given to get reimbursement and to sort of factor into the discussion fairly, and then also licensing. We have a big issue with patents in terms of also that cost effectiveness argument. Sure, if you can do the genome for 100, but what if the person who owns your patent is sticking that test at 450 and isn't going to change it? So those are my problems, but we'll wait for solutions.
That's great. And since you finished early, on the question of benefit for reproductive testing, does the experience of Tay-Sachs in particular or other diseases that have been really attacked by different groups and reduced, does that give us tools or precedent for measuring outcomes and benefit?
Well, it's interesting because Tay-Sachs, I think, happened within a community, and it's a very targeted population. Tay-Sachs is a clear benefit. I mean, really, in the United States, it's clearly known. I think there are really no more cases of people who know they have Jewish ancestry getting Tay-Sachs. It's people who don't understand their ancestry or are getting it from a different source. Cystic fibrosis would be the next thing, and that's similar to SMA and through a lot of things that have large, kind of hits all populations, maybe a little differently, but a larger. I was saying during the break, I've spent probably 7 years looking around for articles about what has the benefit been from having widespread carrier screening for CF. I've never seen a study published talking about anything like that. There was an article a few years ago in the American Journal of Medical Association, or the New England Journal, and it was actually about newborn screening. And Massachusetts keeps, I guess, good records and has for a long time, and was also an early adopter of CF screening. In Massachusetts, since, well, the first time they looked at CF, it was down by 50% in newborns. The only way they can attribute that is to screening. But I mean, it was like a footnote. It wasn't even, to me, that's a major finding. But it wasn't treated like that at all. And I think for those sort of white elephant reasons.
Thank you. Pretty hard to measure in terms of costs. I mean, the benefit for an individual family is obvious, but hard to document for a society. Okay, so we'll get you to define a final problem, but for now, I'll put your problem as what's the benefit of reproductive testing, and hope you can offer us at least one solution when we come back around. And Robert, great to hear from you from Veridex.
Thank you. Good afternoon, everyone. My name is Bob McCormick. I'm a scientist. I've spent my entire career working in the device world. I currently work for a small company in New Jersey, Veridex, and we're involved in cancer testing. And I'm going to be speaking from the position of a device manufacturer. Devices rarely get the play that drugs get in the press. And I always like to start presentations out like this with one fact, and that is that diagnostic expenditures account for less than 2% of healthcare costs, yet factor into over 70% of medical decisions. So what we're talking about today is very, very, very important, because I think we've kind of lost our way, and that's going to be kind of the theme to my 5 minutes. I'm going to say that about 2000, in the year 2000, there was a change. Prior to that, the road to commercialization for a new diagnostic technology was pretty clear. As far as a regulatory point of view, you knew exactly what you needed to do, and as far as reimbursement, you also had an understanding of what to do. I'm not saying it was perfect, but every device manufacturer had a clear understanding of what was needed to get their test out onto the market and used into patients. Approximately around 2000, it changed. And I think what changed is the nature of the technology we're talking about today, molecular or genomic technology. It's very, very complex. Genes and the understanding of gene regulation is a learning process. Furthermore, the tests that they are going to be put into are being asked to perform in ways that have never been conceived before. They're being asked to diagnose considerably more complex disorders like cancer, cardiovascular disease, diabetes that are multifactorial. When a patient's being treated, they're not asked to just tell a physician if the patient is doing well in their therapy. They're actually being asked to tell a physician which drug to use. These are very, very difficult questions to address. They require a lot of information. They require a lot of practice. Unfortunately, what has happened because of all of this is that we've seemed to have lost our way as we try to agree on the amount of evidence that is required to reduce these tests to practice. Words like clinical utility have crept into the dialogue as we've gone through all day. We're not going to go through it in any great detail today. But also some things have occurred in reimbursement related to clinical utility that makes it very difficult for manufacturers today. And the whole concept of guideline groups have come into the picture, and unfortunately none of them seem to agree with each other as far as what is required from a test to be used in a patient. So what we need to do is to get back on a clear, straight path to success for these technologies and ultimately, of course, for the patients. We need to determine what is required and how we're going to implement that. As far as the what, I want to focus for a moment on the term clinical utility. Clinical utility, as you know, is your test being used to make a decision on the patient that ultimately benefits that patient. That is the same as a drug trial. In a drug trial, a patient receives a drug, you follow them to outcome. In clinical utility for the device world, a decision is made off of your test and you follow that patient to an outcome. In every sense of the word, it is exactly like a drug trial. Today, no diagnostic company is prepared to commit and successfully deliver that type of a study simply because it's never, ever been part of the fabric of diagnostic testing. So we need to come to terms with that. What makes it worse is, as you know, the agency does not necessarily require clinical utility. So instead of having a 40-yard dash to the finish line, it now becomes a race with hurdles, as you have more than one hurdle to get across before you get to that finish line. Third-party payers and clinical guidelines need to agree on exactly what they need for these diagnostic tests and if it is clinical utility. I do not believe that every diagnostic test requires clinical utility, but we need to determine that and we need to implement that in a clear, very clear fashion. So how do we get there? First of all, it can't be done overnight. We cannot say effective immediately you need to do this. It takes many, many years to queue these things up and to pull them off. Okay, I could do that. I will.
If you can restate your problem though,
okay,
before we leave.
So the problem is how do we get back to a road that's straight and clear for all device manufacturers so that we can deliver these very, very beneficial tests for the advantages of both patients and clinicians? Okay?
Awesome, thank you. And I'm going to take the same privilege to tell you about my problem. And it's interesting that you raised the question, Bob, of information as a pharmaceutical, because that was exactly the premise of what got me started in this whole area 11 years ago when we started the REVEAL study, where we said that information is exactly like a drug, and we did a randomized clinical trial of the use of information. And we've been building on that for the last 11 years, looking at this ApoE, risk of Alzheimer's disease paradigm, as a model for understanding different issues in translational genomics. We've also moved on to a survey that we're about to conduct of direct-to-consumer companies, because using this as a kind of model for the discovery of incidental findings. So you go to a direct-to-consumer company and maybe you're curious, maybe you have a particular interest, but you get back, whether you want it or not, a whole lot of different information, and much of it is going to surprise you. And so this is one type of naturalistic experiment that's going on, call it a phase 4 study before a phase 3 was ever done, in the use of incidental information by literally hundreds of thousands of people. But what I'm most excited about is the recent funding we just received from NHGRI to become the first center to actually use whole genome sequencing in the practice of medicine in a clinical trial format. George Church has told you about the PGP-10, where they have actually sequenced people and put their information online. And we have the MedSeq project with apologies and approval of Les Biesecker for ripping off part of his name. And the MedSeq project is going to disclose whole genome sequencing to both specialists about their specialty area and primary care docs about the entire possibility of disease risk in their patients. And we're going to do this as a exploratory clinical trial with an implementation arm and a placebo arm who does not get the same information. My problem, as we struggle to now put this protocol together, my problem is what is an incidental finding and what is population screening? Are they the same thing, as Mark and others have alluded to, or are they somehow different? And the different analogies are informative. The one that most people like to draw on is the imaging analogy, where when the radiologist throws the chest x-ray up and they look at the heart, they have an obligation to look at the lung, and they have an obligation to tell you if they see a schmutz in the lung, very technical term, if they see one in the lung, so that something can be done about it. And they're absolutely liable if they don't. So my problem is, as this rolls out, and I don't quite believe, David Ledbetter, that labs will be able to content themselves with reporting on a limited number of variants. As this rolls out, how in the world does the laboratory deal with the implication that they should look for and possibly report perhaps an incredibly large set of incidental findings? And how does an insurer come to grips with the question of whether this is a standard part of reading our genome, or is this somehow population screening that they don't want to pay for? And I actually have a solution. But we now, let me summarize and ask each panelist for their solution. So Greg, your problem was too many guidelines for evidence-based practice. Please correct me if I've got it wrong. What is your solution?
So I didn't actually mean to imply that there were too many guidelines in genetics and genomics for evidence-based practice at this point in time. That is definitely not the case. But I think there is value in carefully thinking about the evolution of guidelines in this space preemptively. So my proposed solution is that before we get too far down the path, we think about ways of essentially setting a process up for arbitration between conflicting guidelines as they emerge or as they are about to emerge, whereby medical specialty societies and perhaps payers could come to the table as stakeholders and essentially in a structured way duke out the process to get to the point where there is a sort of a unified view on a particular application. And that may help with his issues as well.
Outstanding. And it's too, I think Norman Kahn may have had to leave, but he would be in a position to really do something like that through the Council of Medical Specialties. Okay, Thomas, I think I stated your problem before about CMA and amnio and information of uncertain significance, kind of related to mine.
Exactly.
Help me out. What's your solution?
There are several parts to the solution, but I'll try to outline them very quickly. So one possibility, although this is controversial and to some may seem paternalistic, is to really limit the arrays to those CNVs that have robust clinical data associated with them. A balance between the yield and the complexity of the counseling and decision making is really sort of what we're aiming for. Or to bring up slowly the complexity as we gain more and more, as we evolve the databases and gain much more robust information, and have periodic update of arrays. That involves some technical and commercial risk, of course. To encourage registry entries, which are not great, but it's a place to start collecting data. To encourage outcome studies, not only outcome studies, but utility data and cost-effectiveness analysis on how is this information being used and what is the experience of both patients and practitioners to this period of uncertainty that we catapult them into during these very sensitive weeks of pregnancy. And then lastly, I think education, both education to prenatal care providers with clear guidelines and have people in professional organizations come together in many of the ways that Greg was outlining so we can have some agreement and not have dissent among the voices.
Terrific. Now, we do want you to offer your solution, so, jot down your favorite problem and please get ready to come up to the mics with your solutions. Deborah, you told us that you really wanted to measure the benefit of carrier screening. Have you got a solution to help us do that?
I sort of had two. One was first to create a pathway to actually evaluate genetic tests. And I think that what's been done for newborn screening could be something that we could really benefit from in the genetic testing community. And I sort of have that as a three-pronged test. The first I think we need to do is label the use of the testing that we're talking about, because reproductive planning testing is going to look very different. The evidence base is going to look very different than assessing treatment options or diagnosing adult onset disease or recreational uses for genetic testing. So I think the first is sort of to label what the testing is actually being used for. And then I think it's really to create a criteria for evaluation, which sort of already exists when we look at carrier screening. They usually look at, is the disease significant and to impair health in the offspring? Is there a high frequency of carriers? Is there a technically valid screening method? Is there fetal testing or follow-up possibilities? And is it voluntary? So I'm sure we could come up with some sort of criteria in every group And then I think the third thing is to create a cohesive board that probably has molecular pathologists, that has OBGYNs, that has geneticists, that has neurologists, that has different people who are able to see this from different angles and possibly include patient groups or patients to get some of what that value is worth to that family living with that disease or that person. And then I think the fourth thing we really need are steps to implementation. It's great to have it, but we have to get it from here to actually into a clinic. So we need to have lab guidelines and things like that set up. We need to have physician education, and we need to have some sort of patient education. So I think that sort of looks at, how to evaluate genetic tests. And then I think in terms of the cost effectiveness, the patent issue, I know we've been very proactive in the BRCA case with the ACLU, trying to work on, what, at least for some of us, what a strangling effect the patent is having on access to testing and cost of testing.
Great, thank you. And Bob, how are we going to figure out what is the bar for approval of diagnostic tests so they can be integrated in the marketplace?
Yeah, so as I left off, I think there are three things that we need to do to get this road clear to us all. Number one, again, we need a bridge for those tests that are being introduced now or recently. With higher standards, we need some way to bridge them into those higher standards because it takes time to queue these types of studies up and deliver this kind of information. The second thing is I think that the agency needs to make a very definitive and clear regulatory pathway for the commercialization of these tests and to define very clearly for us the role, if any, of LDTs in this laboratory-developed test I refer to here. The third is probably the most important, and if we are going to begin to equate some devices with drugs, then we need to go all of the way, and we need to affect a mechanism where one study will suffice for the needs of all the important stakeholders. That one study will give you regulatory approval, it will give you reimbursement, and it will give you guideline inclusion. And I believe that is the biggest thing we need to work on.
Oh, that's going to stir some discussion. And if you'd like to start lining up at the mics, please do so. I'm going to say that my problem was the interface between incidental findings as a kind of part of diagnostic testing and population screening, which reimbursement entities don't want to pay for. And so my solution is a minimum list. It's very low tech. It's getting a minimum list which can be incrementally changed over the years of variants or conditions that at least hopefully a consortium of bodies agree should be done by each lab if they choose to seek and report incidental findings. And I imagine in my head that list to be less than 25 entities right now, perhaps, but we're actually working on that for the ACMG. So, fantastic presentation. I'll ask you, people coming up to the mics, you may speak about anything you want, of course, but it might be most on task, since we've been tasked, if you can pick one of these problems and actually try to address it with a new solution. And I will ask you to keep your remarks to about 1 minute or less so we have enough time for other people who want to speak.
Okay, so I don't have a solution, but I did want to echo the issue, the regulatory issue that was brought up by Robert McCormick. For instance, right now with BRCA testing, people are using it to make surgical decisions. The test is not FDA approved and was not ever required to be. Recently, there has been development of drugs that may beneficially work on people with BRCA mutations. And when we met with the FDA, we were told that BRCA analysis, the test, cannot be used as a companion diagnostic for pharmaceutical companies to develop these drugs and test these drugs in clinical trials because BRCA testing is not FDA approved. So it's a little ironic that people can use it to make decisions about surgical prevention and that it's not considered a, I guess, good enough test, or that the regulatory process was not good enough to allow it to be used as a companion diagnostic test for a drug.
Bob, any response?
No, other than I agree with the confusion. Absolutely.
Excellent. Mark?
I'm going to offer comments to two, but very briefly, a friendly amendment to Greg's to bring something back into the conversation that I think Dan mentioned earlier, and that is the issue with guidelines relating to them being not adequately explicit. As my friend Alan Morris says, they're loaded with weasel words. Consider this, could do this, might consider. That's what the guidelines look like, which are really useless. And so if we really believe something, we need to state it in such a way that it's explicit what the provider should do with the information, and then that can go forward and be computed into a clinical decision support. I would also note as a nuance to that there are some groups that are looking at these types of guidelines and or algorithms as a proprietary, and there may be intellectual property issues that are going to crop up here too, which would be disastrous in my opinion. So I think that needs to be added. And then to Thomas's point, to reinforce what was brought up before, I think we could develop a clearer transition between the clinical and the research side. We're going to be implementing a whole genome sequencing process. We're going to look at what we know clinically relates to the indication, but then we have a clear path to offer the parents and patient to a research arm for things that we find that we don't know what they mean. And that was exactly what the SET program with ultra-rare diseases had done, was to basically lower the barriers so that people could move from clinical into research to try and more rapidly improve knowledge. So that would be a model that I would like to implement as well.
Thank you. Just to clarify, it's 1 minute per speaker, not 1 minute per problem. You were good. You were good. Greg, since he addressed first your thing, he wanted to refine that. You're okay on that?
No, I agree entirely. And I think the point about making the guidelines computable is something that all guideline generating organizations should be thinking more and more about now as we move into a time of CDS.
And anyone else to?
Yes, and I agree with. Thank you for that solution.
All right. Fantastic.
All right, 60 seconds. Federico Goodsid from Vertex. This is for Bob. Bob, the problem that you bring up, if you have a self-standing diagnostic, I understand that the issue of clinical utility really becomes a problem. But as long as we talk about companion diagnostics, isn't the biggest problem right now the fact that perhaps we're putting a burden on the diagnostic that really has no business being there? The clinical utility is shown in the pivotal trial for the drug, where the diagnostic is essential to be able to do the trial. But you're not showing clinical utility for the diagnostic itself. It's for the whole package.
Yes,
together.
I totally agree. Yes.
And I would
As
also
a companion diagnostic, yes.
I was to just say that I'm sure other people are working on this as well, but because our team had experience in creating clinical trials like this, we've been asked to help with a number of clinical trials that are giving people results of heart disease risk variants, obesity risk variants, diabetes risk variants, and then looking at, exactly as you say, the downstream implementation of what either they or their doctors do, and looking at health outcomes. So trials can be constructed around these concepts, even around something as diffuse as a susceptibility risk variant.
Yeah, and if I can also add, too, I think one of the reasons why device manufacturers were reluctant to tie their test to an outcome is because what happens between their result and that outcome is a lot. And they're really spatial in time, and it's not always related necessarily to the test. So I think this is something that we still are trying to sort out.
Alberto Gutierrez from the agency. I guess that's the FDA.
It's either that or the CIA.
Yeah.
A couple of comments. First, personalized medicine. I'm not sure how you don't tie the results to the diagnostic, because if the diagnostic is wrong, you're going to be giving the drug to the wrong people. And that is, in the end, the crux of the matter. Do you want to give a personalized drug that is geared specifically for a certain population of patients to the wrong people? So having said that, we only do clinical utility, if you wanted to call it that, in those cases where you have a predictive test that is tied to a drug, and usually the drug has to do the studies anyway, so the diagnostic is riding along. For regular diagnostics, that is, if you want to do a prognostic test, we haven't changed in any way what the standards are that you need to know, the kind of data that you need to have to bring those tests into the market. So I disagree with Robert in that a little bit. Those standards are what they have been for the most part.
But may I ask you, I think he's saying it's not that you don't have standards, it's that your standards are not going to be the answer that reimbursers want to hear.
That is correct.
And so do you have a solution to, what should be the answer?
There is right now, and this was available previously, but perhaps nobody really thought of bringing it up, but there is now a collaboration between the FDA and CMS in which we actually are taking one pilot test through this. Exact Science has volunteered to do this, and they are doing a study that will hopefully suffice both for CMS and the FDA.
So there is a model of FDA CMS which you believe would be the bar that might be shot for by these companies who want to get their test on the market.
That's correct.
Awesome.
And one last thing, although we'll talk about laboratory developed testing. But what the agency has said is not that we won't allow laboratory developed tests, but that a test that is, again, a predictive test for a drug needs to come through the agency and get approved. There have been laboratory developed tests that actually have come through the agency and gotten clearance or approval. It's quite possible to do.
While he's at the microphone, does anybody else want to take on the FDA? Okay. Other comments and possible solutions? Well, let's wait till he gets on the panel, and we will definitely bring that back to him. But I have a question, really for Thomas. You mentioned utility studies and outcome studies, sort of like what we're talking about here. But how do we possibly do that when particularly CNVs and copy number variants are so unique and so rare among themselves? How would you ever get enough people or make that happen to do a utility study with a particular given CNV?
Well, I think it would be difficult to do if it was completely relegated to one particular CNV. I think I was speaking more about the general process by which this other, new information has come up, and how, first of all, patients value it, and secondly, what kinds of decisions they make once they receive it, and ultimately what kinds of outcome measures we could put around it to give some credibility to having this be a heavily or strongly recommended diagnostic pathway. So
I think we are almost out of time, but please, one more question.
This is a question for Greg around the guidelines, because I totally agree with you about having more uniformity or of process around the development of guidelines. So how do you address the need for different healthcare provider organizations to feel like the not invented here syndrome? So if we didn't do the guidelines development, it's not applicable to our providers, and to have more cross-cutting guidelines that various healthcare providers can groups will agree to.
That's what those weasel words are for. Unfortunately, medicine or healthcare in general is inherently a tribal endeavor in the United States. I think we need to think about ways to make those tribes feel that they're all one in these endeavors. And I don't have a good answer. I mean, obviously, as soon as there's a financial incentive for one group to go one way and another group to go another, there are going to be huge issues. But we do have to realize that at the end of the day, the people who are with the patients are having to make a decision about which way to jump. I think the alignment of the guideline that comes out with reimbursement is probably the most effective strategy to ensure that you get where you want to be.
All right, well, I think we'll wrap up now. Let's give our panelists a big hand. Thank you so much.
Thank you, and thanks to our moderator, Bob. Good job.